Let me welcome then our chair for the first session of the day, Nicole Soranzo, who's known to many of you. Nicole is the head of the Genomics Research Center at the Human Technopole in Milan, and before that, a longtime human genetics group leader known to many of us at the Sanger Institute and faculty in human genetics at the University of Cambridge School of Clinical Medicine, where she is well known for her cutting-edge work in the genetics of immunology, hematology, and cardiometabolic disease. Nicole?
Thank you, Mark, for the kind introduction. It's really my great pleasure to introduce the next plenary sessions, which is on analytical resources and methods. We have really three outstanding speakers that are shown in this slide. As usual, I would like to remind you that there's an opportunity to ask questions using the ICDAbio 2022 slide. So the first speaker today is Professor Nicola Mulder, who is the head of Computational Biology Division at the University of Cape Town. He is also a leader of the H3A BioNet and the SI Africa Open Data Science Platform. Nicola will give a talk on developing genomics resources for Africa. So welcome, Nicola.
Thanks, Nicole, for the introduction, and I'd like to thank the organizers for the invitation to speak. So I'm going to talk a little bit about why we need African-specific genomics resources. I know there was a big discussion yesterday about not creating these in silos, but I'm going to give a little bit of argument for why we're doing this with the hope that eventually these will converge on the public resources. And then I'm going to talk about some of the specific resources that we have, particularly as it relates to the M2M within the ICDA recommendations. And if there's time, I'll talk about an open data science platform we've just started working on. So I'm not going to go through the bits in italics because it was discussed many times yesterday about the importance and the huge diversity of African genomics. So I'll talk a bit about the challenges. We also heard yesterday that African genomic data are very rare. It's very hard to find them. There's a lot of reluctance to share these from African researchers in global public resources. When they do exist, they're really not easily findable or accessible, and the data are not represented at a granular level. So I'm just going to talk a little bit about this paper that was published that we worked on a couple of years ago, and it really argues for why we need access to the data, but also why we need access to the data at a granular level. So this data set, this project was an African-led analysis of 426 individuals from 50 ethno-linguistic groups across Africa, and you can see the PCA for that. So they it covers some of the four sort of major language families, but you can see that in the PCA they would split according to your geographical distribution. But there's also individual splits within a country. For example, in Uganda, you can see distinct splits of clusters within this country of different population groups. So the paper also identified more than 3 million novel variants that have these novel as in not existing in current databases. More than 80% of these were singletons in a given population. And you can see here on the left, the novel variant discovery for all variants continues to increase as we add new populations. And even if you look purely at the common variants, there's still 6,000 variants between the last two populations. And the important thing to note here is the regional variation. So there is individual and regional variation in the number of novel variants, and this is very much correlated with the ancestry of the individuals. We also looked at highly differentiated variants between populations. So these are variants that have at least a 0.4 difference in frequency. And there were 11,500, 3/4 of which were in the MHC region or around the sickle cell locus. But there were over nearly 2,500 that were not in those regions. 10% of which focused on hits, were near GWAS hits. So again, this regional variation across populations, and the same is true for loss of function variants. So the red bars on this graph show those that are common across the different populations that we looked at. And you can see the regional variation as well. And I'm not showing the results here, but the signals of selection also showed distinct regional variations. When it comes to the medically relevant variation in African populations, there were very few hits to the ACMG list because it's been designed for particularly European populations. But there were a lot of hits to likely pathogenic variants in ClinVar. And these are individuals walking around with reasonable number of these hits. And again, massive variation or reasonable variation in the frequency across different populations across the continent. So if we look at just some specific examples, so if you look at the sickle cell mutation, so this is the orange bar across here, you see it's very much endemic to the malaria regions. In the south, you hardly see that. When you're looking at additional variations across in one particular country, so this is the Uganda example again, you can see that these variations that are of African interest very, very, very much in their frequency within this particular country. And this comes back to that PCA where you could see these distinct clusters. And it's because of the influence on the Nilo-Saharan cluster on the right from North African migration. And so it's important that when you're looking at medically relevant variation and actual implementations, that it's essential not only to look at sort of geographical regions like Northeast, Southwest, but even within a country to look at what is the ancestry of different population groups that may be relevant for genomic medicine, for example. So the study really highlighted the granularity is very important for considering regional variation. We need more African data. This is a reasonably small geographical distribution and data sets. And we also obviously ideally need some more clinical and phenotype data associated with that. This was pure implications from the frequency data itself. And the data are globally relevant. So we've had, within an hour of the paper being published, we had the first request for access to that data set. Often people just want the frequency data. So this is why we have ended up having to develop a number of African genomics resources. And when I looked through the recommendations on the white paper, a lot of these relate actually to some specific areas, to the genomics resources, obviously. Here I'm going to talk about our array, invitation service, and reference graph. And then data portals. So we've developed a couple of portals, and if there's time, I'll talk about the new the Open Data Science platform. But of course, there's something I don't have time to talk about, which we've invested enormous effort into, is training in genomic medicine in all areas of genomics. So firstly, in terms of data generation resources, so we, several years back, we designed the H2Africa array. This is a genotyping array specifically for the H2Africa Consortium. It has reasonable proportion of fixed content, Illumina bead pools, but we were able to identify pools which were enriched for African-specific variation, and then we were able to add custom content to that. So we got specific requests from the PIs who were working on specific diseases. We then got ones that weren't already in the bead pools from the main databases for which there would be hits in African populations, and then GWAS catalog and others. And then we also included some mitochondrial Y and ancestry informative markers. So this chip had about 2.4 million SNPs. We evaluated this chip compared to a number of others, including the other Omnis and the mega chip. And there were distinct differences, improvements actually in our chip. The red lines at the top are ours. One is the final list, and the other one is the one that actually passed all the QC and is actually on the bead pool and it passed manufacture, etc. And they all sit above the other arrays because they're obviously designed to improve the imputation in African populations. And the same was true for the coverage. We looked at coverage across all different regions and for each different African regional area, geographical location. And you can see that the smaller chips like the Mega and even the GSA, we haven't got those results on here. They don't perform very well compared to the bigger chips because of the shorter LD blocks and the diversity. So the array, we've just, about a year or 2 years ago, actually had V2, where we added another bead pool for additional SNPs that have come out of H2Africa. And an iScan has been placed in CPGR, so we can now generate the data in Africa. And over 50,000 participants have been generated within the consortium on this, have been genotyped on the consortium array. And what's interesting is that many of these studies, they've reproduced some previous SNPs, or they've reproduced the gene, but it's a novel SNP. And there's some that are identified SNPs that are novel to Africans that would not have been found with other chips and potentially other reference panels. One example is this paper from the OurGen project, which said that the use of this African-centric array enriched and the enriched African reference panel enabled them to capture diversity around the two loci that were found. It's really important that the right data generation of tools are used for our populations. We provide additional resources associated with this array. So the manifest files, the cluster files, and then we have a website where you can browse the different, search the different markers on the array, and browse them, view them in a genome browser to see where they are located. The other thing we provide is a reference panel and imputation service. Now, we know that many reference panels exist, and the bigger the panel, the better, but we were sitting with this data set that was also published in the Nature paper, which was under controlled access and not allowed to be released in a reference panel. So we created a reference panel from the data set that was used to design the chip, and then we've added a whole lot of additional whole genome sequence data, again, data that's in the public domain, but also data that people are not willing to share. And we've got our imputation service uses the open source Michigan imputation service front end, so that people can come in and run without actually having access to the Extra African reference panel. They can actually run their imputation on whatever chips they're working on. So we've tried to assess the imputation panels and different methods, because we know that there obviously are differences. I'm not talking about any of the results from the full imputation. We're about to write this up for a paper. We've also, one of my students recently just did an analysis of the MHC region, looking at general imputation tools as well as HLA-specific tools, and looking at these in the light of different reference panels and different chips. So this bar chart shows the first two are the H3 Africa chip with the first one's the H3 Africa chip, which with the different methods, and the other is the Omni chip, and you can see their distinct differences. And then looking at a number of well-imputed SNPs for different reference panels. And again, the differences in the different reference panels, particularly at the rare variant level. So ideally, we need federated imputation. I've come across several other examples where people have private data sets that can't make the imputation panel available. But wouldn't it be nice if people could just run their imputation at multiple different places and have a way in a federated manner, and then have a way to nicely merge those results? So that's something we're looking forward to having. And then when it comes to reference genomes, we've all heard that the current linear reference genome is biased, and it doesn't capture that broad diversity in Africa. And so we need a more representative one. I mean, the paper from Sherman et al. A few years ago identified at least 10% of the African pan-genome that is missing in the current reference, and this is using African-American samples. So imagine if we use a huge diversity of African samples, how much we're going to identify that's not represented in the reference genome. So we've got a project that's looking at both a pan-genome and a reference graph. So for the pan-genome, we're in a small pilot, we're looking at just over 400 individuals from, we basically selected the main ethno-linguistic groups, or the language groups, and looking at PCAs to see where we have reasonable coverage across the continent of different clusters. And then got those sequences and run them through the HuPan pipeline. Now, again, we're going to be sort of pushing these results through soon, but the preliminary results are showing many megabases of novel sequences, significant contribution, which I think is not unexpected from the Khoisan ancestry, and a lot of regional differences in contributions to the pan-genome. So we're also then taking these sequences plus some long-read sequences and generating a reference graph. So again, just small data sets, small sample sizes. In Africa, we just don't have money to run massive sample sizes, especially with the expensive long-read sequencing. So we're just generating hi-fi data for about 30 different individuals, covering these 5 different regions on the PCA. And we're running we've been busy benchmarking various different methods, and we've got 2 hi-fi genomes done through a local competition. We won 2 free long-read sequence genomes, but the samples are ready for submission now for this sequencing. We had a lot of issues with the consent, because we're asking for as open consent as possible, because we need this reference graph to be openly available. It's not as open as the Human Pan Genome Reference Consortium would like, but we have to take small steps in consenting and open access and African data. So we've had a lot of discussions with the HPRC to work with them on aligning our methods such that we can have our reference graph contributing to theirs, or at least as a supplement to the main human reference. So that is some of the tools. We also, as I said, have been creating a couple of data portals. One is to address the issue of finding data in existing public resources. So this is the African Genomic Medicine portal we've created, and that retrieves existing data, particularly genotype to phenotype data. And then another one is the African Genome Variation Database, which addresses the issue of accessing summary data at a granular level, particularly for data sets that are not openly available. So the AGMP looks at from GKB, does GNet, and soon, we haven't got the data in yet, but soon to come is the GWAS catalog data. And basically just extracting the African-specific data from these databases, actually taking the papers, reading the papers, curating and extracting additional information from the papers. So we've created this portal where you can search by disease, drug, variant, or gene. Efavirenz, for example, is an antiretroviral that's relevant to African populations, particularly the variants in there. And then we provide the variant drug associations that all links directly to those public databases. But the pieces that we add are, p-values if we can find them in the literature, and we break the data down into, we go and find what actual population it was, and we break down into a more granular level. So then what if we want to find out more for one of these variants about their frequency, and that data is not available, for example, in Nomad, so because Nomad takes high coverage? What if it's, came from an imputed array, or has from exome sequencing or low coverage sequencing? So African Genome Variation Database, what we tried to do here is pull in data that we tried to get a lot of these data owners to submit to Nomad, but there's a lot of hesitation. I think they want to sort of try through an African-owned resource first and then share. So here we make very similar to Nomad, variant results available, but population frequencies available at the granular level, so at the country level or even the population level. And we provide access to the results directly to Nomad, so we don't try and replicate what's in there already. And then there's a world map view where you can view your frequencies. So the idea of this, where the rare diseases community in East Africa has been wanting access to this for filtering their variants for when they do NGS. They have to filter and remove common variants and then do variant annotation. And HEV does this already. We have all the variants, we've annotated them, we've got the frequencies, and so we can do this filtering for them. And we're providing access to beacon networks. So we have beacon search from HEVD, and we also have made the Extra Africa data available as a beacon. So those are the resources. And then I just want to mention this because a data science platform was mentioned in the recommendation. So we've just received a grant from the NIH in the Harnessing Data Science for Health Discovery and Innovation in Africa, otherwise known as DSR Africa, initiative to develop an open data science platform. It's built on the data biosphere concept, so that's Terra and Gen 3, and uses GA4GH standards. And the idea is to have all the cohorts from this consortium available in here, where you can search data, apply tools, and then run on various clouds that are that the African scientists are comfortable with running. So we're trying to ease the barrier to access to sharing platforms and enable federated data analysis. So that's just some of the ways that we have been sort of addressing some of the goals, actually, of the recommendations of ICDA, enabling availability, improving availability, accessibility, and sharing of African data. But we need a lot more. We'd love to create a genotype-phenotype resource as well. We'd love to get some more data, and eventually to merge this with the public repositories as well, so we don't work in a silo. So with that, I'd like to just acknowledge the consortium, and thanks again for your attention.
Thank you so much. This was a really great talk and lots of information. So we have very little time. There is one question, and I would ask everybody else to ask questions through the platform. So the question that was posed by Joelle for this day is whether this African imputation panel is better than TopMed, presumably for African populations.
No, so it's never going to be as good as TopMed because of the size. So the accuracy is very much dependent on the size of the panel and obviously the diversity of the panel. So there will be some cases, maybe for some variants, where it'll identify variants that would not be identified in the TopMed panel. So it's got, I think it's one that needs to be used together with one of the bigger, sort of more powerful panels. But simply, there's no doubt that size matters in this case.
Thank you so much. I'm afraid we'll have to move on. Thank you again. So the next speaker in this session is Joelle Mbatchou. Joelle is a senior statistical geneticist at Regenerogenetics Center in the US, and she works on the development of statistical and computational tools for associations. association mapping, particularly in larger biobanks. So Joelle, we're delighted to have you here. Please to listen to your talk.
Good morning, everyone, and good afternoon to those of you who are on the other side of the Atlantic. I'm Joelle Mbatchou, and I first want to thank the conference organizers for inviting me here today. So I'll be presenting a computationally efficient tool that was developed at the Regeneron Genetics Center for large-scale association analysis. So here at the Regeneron Genetics Center, we gather a large amount of genetic as well as phenotypic data obtained through worldwide collaborations in order to drive target discovery, as well as identify novel indications to down the line improve on patients' health and outcomes. So many methods have been developed to analyze data at this large scale, and they can usually be classified into whole genome regression model-based methods or mixed model-based methods. And they can usually be broken down into a two-step approach. So in the first step, you would fit a genome-wide model using a smaller set of variants, for example, directly genotype variants, and you would use a linear model for quantitative traits or a logistic model for binary traits. And after running this step one, you would test a larger set of variants for association in a step two, where you would be conditioning on the genetic predictions from the first step using a leave one chromosome out approach to avoid proximal contamination. And an example of a widely used method for quantitative traits is bold LMM, and for binary traits is SAGE. And so while these methods were developed to analyze data at this scale, they still remain computationally burdensome when you think of the large amount of phenotypic data that's being derived, as well as the growing size of the data sets, both in terms of samples, but also in terms of the variants that are being tested. And so this really brought forth the need for us to develop a method that was more scalable when you take into account all of these factors. And so that's where we proposed Regini as a way to address these limitations. So Regini was developed to work on both quantitative as well as binary traits, including binary traits where you have a high amount of case control imbalance, where we use penalized first regression as well as saddle point approximation to avoid inflation due to that imbalance. Regini also controls for population structure and genetic relatedness through the use of a whole genome regression framework, which I'll go into in the next slide. So Regini uses efficient algorithm to process multiple phenotypes within the same run. And we've designed it to decouple the whole genome regression step from the association testing step. And I'll go into examples of why that can be convenient. And so we've made Regini publicly available on GitHub, where you can use it on your data sets. So going over the whole genome regression step in Regini, so the key idea at this step is when we took into account the large size of the genetic data that was to be used in the model, so both in terms of the number of samples, but also in terms of the number of variants that are used. When we ingest this genetic data, instead of ingesting the whole data set all at once, what we do is that we only ingest it into smaller blocks, where for each block of variant, so we read in that block, and we summarize the information in that block using a method that's called ridge regression. And that method is has close ties to the infinitesimal model that's used in linear mixed models. And so for each block of variants, we extract rich predictors so that when we fit now our genome-wide model, instead of having this large amount of variants that we include, now we have a smaller set consisting of our rich predictions. And so at this step, we fit another round of rich regressions, again, because the rich predictors are going to be correlated with each other, but also correlate with the phenotype. And so we also apply cross-validation in order to avoid model overfitting. So once we're done with the second round of rich regression, we extract our leave-one chromosomal predictions, which we'll then use in the subsequent step where we test for association using a larger set of variants, whether it's imputed variants or exome variants, for example. So now I'll go into some of the applications of Regini, comparing it to Bolt LMM for quantitative traits and then to SAGE for binary trait. And this can be found in the Regini paper. And so when we focused on the first step, which is fitting this genome-wide model, so we found that with Regini, we were able to reduce the computational time by over 170 times for binary traits and even more for quantitative traits. And then when we move on to the association testing step, while the difference is not as drastic, we were still able to gain into some reduction, both for binary and quantitative traits. And so some of the features in Regini, which help to gain all of this computational efficiency. So the first one is that the Regini framework is highly parallel in nature. So both in terms of the ridge regressions that we use in the first step, or we can reuse some of the matrix operations that are the same for all phenotypes. We've also developed an efficient approximate FERT test, which is able to reduce the timing compared to the exact FERT test by over 60 times. And we actually found that the parameter estimates using the FERT correction were more accurate than those using a saddle point approximation. And that was really important when you were testing at rarer variants or when you had highly imbalanced binary traits. So more recently, we've made many improvements to the software. And so in this slide, we're comparing the initial version of Regini, which was used in the paper, to the most recent version of Regini, which we named 3.0. And so comparing these two versions, so in step one, we were able to decrease the computational time by over 10 times for quantitative traits and 18 times for binary trait. And then in step two, we're able to reduce the timing by 6 times, both for quantitative as well as binary traits. And some of the new features that we've added since the initial version of the software. So the first one is that we've made more efficient use of multi-threading when reading the genetic files. And that was both relevant in step one and also and more so in step two, where we're testing at millions of variants. We've also made use of the Intel math kernel libraries, which help speed up many of the matrix operations, and that was quite relevant for step one. We've made more efficient matrix operations, as well as improved the algorithms that we used. So both for the ridge regression as well as for the penalized FERT in step two. And then we've leveraged the sparsity of when we're testing for rare variants, we're leveraging the sparsity both in terms of storage, also in terms of the matrix operations that are performed. And that was quite relevant for step two, where a major portion of the variation that you're testing tends to be rare. So in the publicly released Regini version 2, which is currently available, so a user would run step one, where they would extract these local predictions. And then in a subsequent step, they could either test single variants for association with the phenotype, conditioning on these local predictions, or they could perform a gene-based test, more specifically a burden test, where they would test a group of variants together. And they could specify annotation file to also use functional annotations when grouping these variants. And so I just want again to highlight here that because we have decoupled step one, where you can actually store these local predictions on disk, so you can actually reuse the same predictions for those different tests. You wouldn't have to run step one again. So in this new version of Regini, I'm pleased to announce that we've extended the set of tests that you can run in step two. So in addition to the burden test that could be performed for the gene-based test, we've also added a set of variance component tests, and then we've enabled the ability to perform interaction tests. as well as to perform conditional analysis, where you would be specifying a set of genetic variants to add to the set of covariates when you're testing variants for associations. So for today, I'll go into the gene-based test feature as well as the interaction testing feature. So with the new set of gene-based tests, the key drive behind extending this feature is that the burden tests are quite powerful when you have variants that indeed have effects in the same direction. However, you can have quite significant power loss when that assumption doesn't hold. And so we first implemented SCAT and SCAT-O, which are variance component tests, which are robust to that assumption of effects being in the same direction. In addition to that, we've implemented tests based on the Cauchy combination method, where key advantages of this approach is that it's highly computationally tractable, but it's also quite powerful when you have few variants in your set that are driving the signal, so more sparser type of architecture. And then finally, we've implemented a novel non-negative least square test, which was developed by my colleague, Andrei Ziadinov. And the idea behind this test is to combine multiple burden masks that, again, are generated using different functional annotations, as well as allele frequency cutoffs. And so this NLS test combines these different burden masks in a joint linear model where it imposes constraints of these masks having the same direction of effect. And so an advantage of this NLS test is that by imposing these constraints, you can actually gain in power when you do have multiple of those burden masks that are causal with the same effect of direction. So if you look on the right, this is an example where we looked at cholesterol level, focusing on the STAB1 gene. And so the set of green points here show the different burden masks that were generated using different allele frequency thresholds, as well as for each threshold, we also use 4 different annotation classes. And then we contrast that to applying SCAT, SCAT-O, as well as ACAT-V. And finally, we also compare that to applying this NLS test, which combines all of these mass signals in this joint model. And so in this example, you can see that the NLS test gives a much stronger signal than all of the other tests, including all of the single individual burden masks. And so this is an example that illustrates the benefit of NLS tests, where it's able to leverage the fact that you have multiple strong burden signals that have concordant effects. So then going on into the interaction testing feature. So the objective here is to test for interaction between genetic variants and environmental risk factors, or to test for interaction between 2 genetic variants, so more for the static effects. And there can be various sources of inflation when you test for this interaction. One main one can be the presence of heteroscedasticity, meaning that your environmental risk factor affects the phenotypic variance of and that wouldn't be accounted for if you just have a model that explains the effect of the risk factor only in the mean. And that's a source of model misspecification. And then to address this, you can use robust or sandwich standard errors. However, these are known to be inflated when you're testing with rare variants. And then finally, another source of inflation can be the presence of high case control imbalance. And so in Regini, we use a hybrid approach to handle all these different sources of inflation. So we make use of these robust sandwich standard error estimators, as well as use heteroscedastic linear models. And so these are models where we can actually incorporate the effect of the risk factor, not just in the mean of the trait, but also in its variance. And then finally, we use penalized first regression, and that's for binary traits to handle this high case control imbalance. So on the right, we show an application of this framework looking at a quantitative trait where we perform a test, an interaction test between genetic variance and an environmental risk factor. And we split the variance based on whether they're common, which is the plots on the top, versus whether they are rare. And if we first look into the common variance, where we contrast the method that uses the robust sandwich standard errors to the hybrid approach that's used in Regini, we can see that those Manhattan plots are pretty much mirror image of one another. However, when we transition now to the rare variation on the bottom, and we compare those two methods again, we see inflation all throughout the board for when you use the robust sandwich standard error estimators, versus when we look into the Regini, which uses this hybrid approach, we don't see that inflation at all. So this is just one example to again highlight the usefulness of this hybrid approach when you want to delve into rare variation. So I hope I was able to get all of you excited about this new version 3.0 of Regini, which is coming out this week. And again, just to summarize key features, so we've extended the set of gene-based tests, moving beyond the burden strategy. We have added this interaction testing feature, so where you can test for both gene by environment as well as gene by gene epistatic effects, where our approach is robust to various sources of inflation. And finally, we've also implemented this conditional analysis feature where you can specify sets of genetic variants to use as covariates when you're running all these tests. So I'd now like to thank all of my colleagues at Regeneron, and especially my colleague Andrei Zyetinov, who developed the non-negativity score test, as well as worked on developing as well as implementing the new set of features that were presented today. Thank you all for your for following. Thank you.
Thank you, Joelle. Thank you for the wonderful talk and also the wonderful work. So there are two questions on Slido, so I'll read the first. The question is asking whether this two-step approach can it be extended to other models like Cox models or inverse binomial?
That's actually a great question. So we have been working on extending the type of traits that we can analyze, so moving beyond quantitative as well as binary traits to also incorporate time to event data. And this is a feature that we have developed, but we are working on more on the implementation into the software and making it as efficient as possible.
Thank you. There is a second question that we don't have time to take, but if you could answer in the slide, that would be wonderful. Thank you again for the great presentation. Okay, so it's now my really great pleasure to introduce our third speaker. It was actually supposed to our second speaker, and that's due to so thank you for staying on. Professor Jian Yang is a legend in the field of statistical genetics. He is now a professor of statistical genetics at the School of Life Sciences at Westlake University in China. And he took this appointment recently after 12 years spent at Queensland QIMR, Berghofer Medical Research Institute in Australia. So Professor Yang has developed many of the methods that we've all been using as a community, and we're really looking forward to listen to his talk, which is on understanding the molecular mechanism underlying the polygenic architecture of human complex traits. Thank you and welcome.
Oh, thank you, Nicole, for the kind introduction, and thanks the organizer for the opportunity to talk today. So I will be talking about a few methods or tools that may be helpful for us to understand the mechanism underlying the polygenic architecture of complex traits. I guess we all agree that most human traits are complex, and years of research in this field may boil down to a single statement that all common trait and disease are affected by a large number of variants with small effect, and the so-called polygenic model. So I don't think I have time to expand the work that we and others have done to understand the cause of the polygenicity. And as far as I understand, there are at least two factors that may be the cause. One is a large number of mutational targets underlying a trait, and the other factor is the action of negative selection or purifying selection acted on new mutations that are often deleterious. So with this polygenicity, a large sample size is essential, and of course we understand this well. But that large sample size also brings challenges for us when we try to analyze large data set from, for example, from the biobanks. And I think Joelle has done a great work in introducing the model. That makes my talk a lot easier. So in a traditional linear mixed models, we often have this kind of polygenic term modeled by a genetic relationship matrix to control for population stratification and relatedness. And that under this setting is often computationally challenging to solve a mixed linear model, in particular in association analysis that involve millions of genetic markers. We so kind of contribute to this field by introducing this method called FastGWA. So we took a very kind of classical strategy. So we use principal components derived from genetic data to control for population stratification, and a sparse genetic relationship matrix or a pedigree relationship matrix to control for relatedness. And because this relationship matrix is often very sparse, and we that allow us to develop very efficient sparse matrix-based algorithms, and together with efficient computer code to speed up the analysis by orders of magnitude. So here, just example, when we applied FastGWA to the UK biobank data with 400,000 people, it runs the total runtime for a single G was about a quarter hour, 15 minutes, and with very reasonable memory usage. Well, FastGWA can be applied to binary traits, but as Joelle has already mentioned, when the binary phenotypes have low prevalence, the test statistics can be inflated, in particular for rare variants. So we have extended FastGWA under the generalized linear mixed model framework. And again, we use PCs to control for population stratification and a sparse relationship matrix to control for relatedness. And we call it FastGWA GLMM or FastGWA-B. We have challenged the two tools by a super large simulated GWAS data set. Essentially, we just duplicated the UK biobank data many times until the sample size reached to 2 million. And that pseudo GWAS data allows us to test the speed of the two methods. So as you can see on this graph, even with a sample size of 2 million, you can run a GWAS with 8 CPUs within like, I guess, 10 hours for binary traits, for quantitative traits, and for a binary trait with FastGWA-B, it can be finished within a day. Again, the memory usage is very reasonable. And we have applied a FastGWA and a FastGWA-B to about 3,000 traits in the UK biobank and have made all the summary statistics available at FastGWA data portal, which is developed based on feedback. So it has all the feedback features like the Manhattan plot for each trait, a locus plot, and a fieldwork plot for a particular variant of interest. And of course, the for the ICDA mission end-to-end, we know now that the bottleneck now is how can we translate the GWAS findings to biology, and here I just want to show you just one example to show how difficult it is just to identify a single gene underlying a association signal based on a traditional model system, and the results are often inconsistent and take a long time. A few years ago, we contributed to a method, one called SMR, which is developed under the Mendelian randomization framework to estimate or test the effect size of transcription on a phenotype using or integrating EQTL and GWAS data. And we also developed a method called HIDE to distinguish whether the association between the expression level and a phenotype is due to a set of shared causal variants underlying the association signals, or whether the association between gene expression and trait is driven by genetic variants in LD. But I guess one mechanism that is less well studied or often underexplored is alternative splicing. Despite that, roughly 95% of multi-exonic genes are alternatively spliced. So we were wondering what's the role of alternative splicing in mediating the genetic effects for complex traits. So here's just a cartoon to show the effect of a genetic variant on splicing. So in this example, a genetic variant can affect the splicing of this axon, and it changes the expression level or the abundance of different isoforms, but it doesn't affect the overall expression level. So as you can see, that if we run association analysis to do an EQTL test, there's no effect. But in fact, this it shows a strong effect on both isoforms with opposite directions. And of course, this is just one extreme examples, and more often we see examples like this, that a genetic variance is associated with the overall expression level and can also affect the splicing of the gene. So we have recently developed a method called CISO to run a splicing QTL analysis. So the basic idea behind this method is to test the heterogeneity in the effect size of the isoform level EQTLs, and because this heterogeneity is a testing among estimates that may be correlated, because isoforms can be correlated, and we just run a multivariate test, acknowledging that the differences are correlated, and we use a set point approximation to do a sum chi-square test, which is essentially a method very similar to that implemented in HIDE test. And we have applied this method to brain data sets, brain and RNA-seq data sets with genotype data. This is essentially the data set, the largest data set available of this kind in the public domain. So the sample size is about 2,400, and we run a standard EQTL analysis with OSCAR, and then we run splicing or SQTL analysis with CISO and the leaf cutter, because this method can be complementary to each other, because leaf cutter focus on specific splicing events, can be more specific to original or sort of splicing specific to specific introns, while CISO is kind of isotome level test can be more powerful to detect more complex splicing events. So we then integrated the EQTL and SQTL data with GWAS data for a range of brain-related phenotypes, as shown here. So in terms of the relationship between SQTL and EQTLs, we identify a large number of genes with SQTL only or EQTL only, and of course a larger set that are in common with both SQTLs and EQTLs. But for this set, it's surprising that nearly half of them show distinct EQTL and SQTL signals, as shown here in the co-local analysis. If we do a co-localization analysis between SQTL and EQTL signals, a large proportion of them show a high sort of posterior probability for model 3, which means that there are two distinct signals driving the co-localization, and of course supported by a large group of the lead SQTL or EQTL SNPs in low LD. Reassuringly, if we look at the enrichment of the SQTL or EQTL signals or the leader SNPs, the SQTL leader SNPs are highly enriched in splicing sites. And when we co-localize the SQTL and EQTL signals with the GWAS data, the number of sort of genes identified through SQTL only is even or higher than those identified through the EQTL data, with about 100 genes in common. That again suggests the important and distinct role of splicing, QTL or alternative splicing in complex trait variation, at least for brain-related phenotypes. Now here, just one single example. Here is association for schizophrenia, which is very likely to be driven for the genetic effect is very likely to be driven by genetic control of alternative splicing for this gene rather than the overall RNA abundance. And with QTL data, EQTL data only, this signal will be missed in the co-localization analysis. And finally, just want to introduce a method that allows for integration of molecular QTL data from multiple multi-omics data. I don't have time to introduce all the method details. Just quickly, that we have applied this method. It's essentially an extension of the SMR method to different omics levels. And here is a application that we collected 5 sort of XQTL data from 5 omics levels, like the protein abundance, iron abundance, DNA methylation, histone modification, and chromatin accessibility. And we integrate all the level 5 levels omics data with the GWAS data for a range of phenotypes with powerful GWAS summary statistics available. So when we look at the effect size of the lead SNP on the molecular phenotypes, we see an interesting attenuation of genetic effect from chromatin marks to histone modification, DNA methylation, gene expression, and to protein abundance. So it seems there's a trend of the attenuation of genetic effect from molecular phenotype away from the DNA, either physically or functionally. And on the other hand, if you look at it, again, there's a sort of increase in sort of polygenicity from chromatin marks to histone histone marks and to DNA methylation, to gene expression, and to protein. Although the trend is not that apparent as a source for the genetic attenuation of the genetic effects. And when we applied this the OPERA method to integrate all the QTL data to all the phenotypes, we observed that although we can find individual cases where the GWAS signal is co-localized with all the sort of XQTL signals studied in this analysis, but by and large, the co-localization are independent across different omics levels, and overall, we identified that about 45 of the GWAS signals are shared at least with at least one XQTL, and accounting for LD, meaning that this sort of sharing or co-localization is likely due to the same codon variant underlying the GWAS and a molecular phenotype. This is a sort of very encouraging result, which kind of suggests that at least a large proportion of the GWAS loci can be explained by the molecular phenotypes, and this number, the proportion, will keep going if we include more levels of data, taking account the spatial temporal effect, and with a larger sample size. And with that, I'd like to acknowledge people in my lab previously at UQ and currently at Westlake University, and particularly those people that contributed to the work that I have talked about today, and my collaborators and data and foundation support. And thank you for attention.
Thank you so much. Again, we have a couple of questions. I would like to ask one now, and perhaps you could answer the latest on online. So this question is from Anonymous. Hi, Jian, great talk. How does fast GWAS GLM compare to Regini in inflation and performance? Also on MR, how does it compare with 2-sample MR?
All right, so I guess the sort of advantage for fast GWAS is that it can really challenge the big sample size, like a few millions for individual traits, while Regini has the advantage to, if you analyze many traits at the same time, it speed up the analysis for the compute time for a single trait, from my understanding. So SMR and 2-sample SMR, 2-sample MR, well, SMR really is a type of sort of 2-sample MR. I guess there's really no essential difference. And I guess the important addition of the SMR package or method is the Heidi approach that utilizes LD to distinguish association due to causality or plurality from linkage or LD between two codovariants.
Thank you so much. I'm afraid we run out of questions because we want to allow for a little bit of time for a 15-minute break. So the second panel on cellular models or M2M2M would reconvene at 2:40 p.m. GMT. So with this, I'd like to really thank again all three speakers for a really wonderful session, and see you all later. Thank you.